Yeah, thank you for the kind invitation for this webinar to present some of our experience. As you said, I'm Jaco Kraan. I'm a postdoctoral researcher in Erasmus Medical Center Cancer Institute in Rotterdam, the Netherlands, working with the cell search already from 2005. And I'd like to share some of our experience with the 4th channel. I would like to shortly present one of our studies with the commercial HER2 marker. I would like to show how we use the 4th marker as an exclusion marker for CD146 enrichment, or MCAM, and some of our recent work using PSMA FITC conjugated antibody in a clinical prostate cancer study. First, one of our studies using the HER2 in the 4th marker. Here we try to investigate the prognostic impact of HER2 positive circulating tumor cells in patients with HER2 negative primary tumors receiving endocrine therapy. And additionally, we also study the prevalence and prognostic significance of these HER2 positive CTCs in a chemotherapy cohort. As said, we included metastatic breast cancer patients with 1 or more detectable CTCs starting a new line of treatment. The CTCs were enumerated using the cell search system and characterized with the HER2 antibody, the HER2 phenotyping reagent. The primary endpoint of the study was the progression-free rate after 6 months of endocrine treatment in HER2 positive versus HER2 negative CTC patients. We used the scoring system very well addressed by the group of Riedorf, published in the Clinical Cancer Research in 2010, where you have a 3+ score with this bright HER2 staining with no background, up to no scoring, a 0 score with no HER2 staining at all. These are the characteristics of our studies we found. We found over the overall 29% patients with HER2 positive CTCs with a staining of 2+ or 3+ or 2+ or more, and the progression-free rate after 6 months did not differ between the endocrine therapy arm and the chemotherapy arm. This is also shown by the Lever-Jennings plots for both the endocrine therapy and the chemotherapy for progression-free survival and overall survival. As you can see, however, we observed a trend in a worse prognosis in patients with HER2 positive CTCs present. We did, however, unfortunately didn't find a significant difference, and therefore we had to conclude that there was frequently occurring HER2 stats between CTCs and the primary tumor, but we couldn't find a prognostic impact in our metastatic breast cancer patients cohort, however. The next thing where we're using the 4th channel for was as an exclusion mark in a study where we used MCAM or CD146 magnetic enrichment on the cell search. And that was initiated by one of our studies where we studied the epithelial adhesion molecule expression on our cohort of breast cancer tumor cell lines. And there we observed that in the normal-like intrinsic subtype cell lines, the EpCAM expression was low or absent. This also here shown in the representative flow cytometry graphs, where you have both the EpCAM expression on basal-like and luminal cell lines, but not on a normal-like cell lines. Therefore, we investigated if we could add additional markers for CTC enrichment using the cell search. And we were looking for candidate markers based on gene expression data, and also showing a high membrane expression on EpCAM negative cells. And of course, there should be absence on hematological cells or easy distinct by additional markers. We came up with 3 potential markers: caveolin, mucin 1, or the melanoma adhesion molecule MCAM or CD146. These were already available from for the cell surge and bound to ferrofluids. And as you can see, this expression of CD146 is very well expressed on almost all of our cell lines, breast cancer cell lines. The CD146 marker is also expressed in endothelial cells, and therefore we use in the 4th channel CD34 as an exclusion marker. So not together with CD45 as been done in the melanoma kit, because the cells are CD146 is also expressed by melanoma cells, smooth muscle cells, but that will be a problem. Dendritic cells and activated T cells, a subset of activated T cells, but we can exclude these with the CD45 exclusion marker. And to exclude endothelial cells, we use CD34 that's highly expressed, as shown here by flow cytometric data, on the endothelial cells here, sorted and positive for Willebrand factor and having the typical endothelial morphology. So in this way, we could exclude circulating endothelial cells using the CD34 in the 4th channel, and make a good distinction between circulating tumor cells and circulating endothelial cells. And we also had the advantage that we could also enumerate the circulating endothelial cells by analyzing on DAPI plus CD34 plus cells. And then you can see that a subset of this endothelial cell also express cytokeratin on a certain level, especially with cytokeratin 8 and 18 is known to be expressed by endothelial cells. This approach was then used in a clinical trial where we combined EpCAM and MCAM cell surge enrichment to increase the CTC capture. That was our goal, and therefore we did 2 countings, one on EpCAM and one on MCAM. We did it before the start of the first treatment and after the first cycle of treatment. These were the results in our study. We had a CTC count of 1 or more CTC in 90% using only EpCAM, 12% in using only MCAM, and combined we had a 25% CTC count greater than 1 or greater, and so we increased the CTC capture from 90 to 25%. And remarkably, 5 patients had no EpCAM positive CTCs, but they did have EpCAM positive CTCs. And after the first cycle of treatment, this was actually increased. We had 90% only EpCAM, 60% only MCAM CTC count, and combined we added up to 30% of CTC count. So here we increased the CTC capture from 90 to 30%. And again, we observed patients with no EpCAM positive CTC, but with MCAM positive CTCs. So we concluded in the study that the majority of normal-like cell lines, so not CTC, but cell lines, do not express EpCAM. They do express CD146 or MCAM, and we slightly increased the CTC detection in stage 2-3 breast cancer patients, combining these approaches. However, it was, of course, we had a low number of CTC positive patients in this cohort with a relatively low, small number of CTCs. So maybe this has a higher impact in the metastatic setting. The last part of my talk, I want to show some recent work where we examined the PSMA, the prostate-specific membrane antigen expression, on circulating tumor cells using a cell surge in prostate cancer patients. PSMA is expressed by normal and malignant prostate epithelial cells, also on the adenocarcinoma cells, and on vasculatures of other malignancies. We first examined the PSMA expression on our cell line cohort, and here you see the very high expression of PSMA on the LN cap cells, and down titrated, and you don't see any background fluorescence in the normal range of 2 to 0.5 microgram per milliliter on white blood cells, and we either didn't see any expression on other non-prostate-like cell lines. Then we did different titration experiments on the cell surge using different concentration of the PSMA, using different times of exposure. And here you can see the expression of the PSMA on spiked LN cap cells. And importantly, of course, we also had our negative controls where we did no stain control, and we didn't see already some small fluorescent background, but the cells are negative here. And importantly, we didn't see any expression on the leukocytes of the PSMA. And now this is a running study where we do use this PSMA marker on patient samples. This is an example of a patient sample where we see CTCs, actually almost all expressing PSMA, only you see some cells with a very dim level of PSMA. Well, actually all EpCAM positive, CD3 to 6 positive cells express PSMA in different levels on the flow cytometry plots. So it looks like the cell surge is not able to detect the low expressing cells very clear. So to finalize, I think the 4 channels can reliably detect antigen markers that are highly expressed by circulating tumor cells. We also have experience with M30, K167, and PD-L1, but I expected this group to be also already addressed by the other speakers. And I think for the future, increasing the sensitivity of this channel, using an other light source, other filter combinations, or a better camera detector, can further improve the detection level of antigen markers on CTCs in the 4th channel. Of course, I'd like to thank my colleagues and co-workers making this all available, and I'd like to thank you for your attentions, and I'm happy to take any questions, and I understood it will be by the chat. Thank you.